Welcome back to Yup Master, dear students, and today's topic is going to be the ribosomes and the intro of cytoskeleton. Before that, we need to know about the similarity and the dissimilarities that we have studied in the previous class about mitochondria and chloroplast. After today's class, you are going to know about that how mitochondria and chloroplast are semi-autonomous cell organelles and how the two are same or different. And then about the ribosome formation, the actual thing about the thing which we used to mention as 70S, 80S, what is S there, and the cytoskeleton, the framework or the supportive structure inside an eukaryotic cell. So let's begin the class with the topics where we left in the last class. I told you that it is better to try yourself. Do it yourself. What? The comparison among the semi-autonomous cell organelles means how they are same and how they are different. So the semi-autonomous cell organelles, as we have studied in the previous class, are two. These two are one mitochondria and the other one that I have wrote in the previous class was chloroplast. Both of the topic, when we concluded, at that time we stated mitochondria as bacterial endosymbiont, chloroplast as bacterial endosymbiont. So there must be certain similarities among the two. But are they totally similar? No, not at all. They are performing different functions. On the basis of different functions, they have some different enzymes as well. Let's start with the similarity first, that how mitochondria and chloroplast are same. So mitochondria and chloroplast as bacterial endosymbionts have certain similarities like a bacterial cell. The first similarity exists in the kind of membrane they possess. The outer membrane in the mitochondria and in chloroplast as well is more permeable. And the inner membrane is less permeable. And the reasons are common. The outer membrane is having porins. Inner membrane is having cardiolipins. Fine. The same way after this membrane is the second thing. What? The second feature. How the two are going to be same. Mitochondria and chloroplast both are same in what kind of things? They have DNA, which is double stranded and circular. Third, both have the site of protein synthesis, means ribosomes. They are same because they are synthesizing most of their enzymes within them, and to synthesize the enzyme they are performing what kind of activity? ATP synthesis is needed because energy is required to make the bond, to make the proteins. And obviously if they have something which is called as DNA, it must undergo self-duplication. The DNA, okay? So I am saying that this is replication. And where these things are present? These are present in their matrix, means the inner chamber. The inner chamber is having these things. Not only this, the DNA is undergoing a process that is called as transcription. Both places, whether mitochondria or chloroplast, their inner chamber is the place of DNA replication. Their inner chamber is the place of translation, protein biosynthesis. So that's why I am saying the protein synthesis or translation requires energy, which is provided by ATP synthesis. Okay. Nextly, it is about increase in the number. The method is called as binary fission. So this is how the two cell organelles or the two kind of cell organelles, mitochondria and chloroplast, are equivalent to bacteria. Okay, and they are similar in all these facts, right? But it is not about the similarity only. There are certain differences. As I said, they differ in their functions. It means they have different kind of enzymes. If they have different type of enzymes, it means the enzymes are participating in different type of metabolic reactions in mitochondria and chloroplast. Let's see how the two become different. These are the similarities. Similarities among mitochondria and chloroplast. The two double membrane bound cell organelle, the two semi-autonomous cell organelles. Okay. The reason of semi-autonomy, I have told these things in the previous classes. We have discussed these things in the previous class about the dissimilar features among mitochondria and chloroplast. And here it is, that how the two are becoming different. Why we are doing this kind of comparisons? The thing is that if the syllabus is finished, say for example, someone asks for the revision of the topic, okay, then how you are going to revise the topic? One of the strategies, complete whole of your syllabus, then start the revision. Okay, obviously because you have studied for whole of the year and you are doing revision at the last, in that cases your retention must be less and you are not able to recall all the things that you have studied in the previous days or those days when you were studying some particular chapter only. So recalling all the features which are similar and dissimilar is not easy for you. The second thing is, do the revision or try to recall or do the comparison at the end of the chapter. Nice, your retention must be much more and you can recall most of the things. The best way is to make the comparison just after completing those topics where you have found certain similarities or some dissimilar features among them. So we are comparing mitochondria and chloroplast just after completing mitochondria and chloroplast because we want to retain most of it, almost everything about mitochondria and chloroplast. Here it is. So we have compared the similarities and here are the things which are going to be dissimilar among the two kind of cell organelles. Now how the two are different? No doubt about this that mitochondria is found in plant as well as animal cell, but chloroplasts are present in plant cells. So this made chloroplast occurrence different. Here it is eukaryotic cell, means either plant or animal. The place of nutrition, the place of function where they are going to perform their function. So mitochondria will perform the function only if the cell is exposed to molecular oxygen. If molecular oxygen is not available, mitochondria is not able to perform its function. The same way the chloroplast can perform the function only if the cell is exposed to sunlight and carbon dioxide availability. That is what required. Remember, the chloroplast, if not exposed to the sunlight, they are called as etioplast, ETIOPLASTS. If you are going to the DPPs, you will find this word etioplast. Etioplast are the chloroplasts which were not exposed to the sunlight, so they are not having chlorophyll in them. The chlorophyll synthesis is dependent on light, right? Remember this. Okay, so we are saying that CO2 and sunlight must be there if chloroplast is supposed to function. In that cases, we say the mitochondria, the place of aerobic respiration, is going to take in what? O2 and give out CO2. So this is how, which is increasing the CO2 content of the surrounding or making the environment more of the gas which is not supporting combustion or respiration. CO2 content increases, fine. Whereas the cell organelle that purifies air is chloroplast. It is taking in CO2, giving out O2. So that's why one who is making the gas impure or the air impure, the chloroplast making the air more pure. Role. What role they are going to perform? As I said, mitochondria is known to perform aerobic respiration. Now aerobic respiration means oxidizing reaction. oxidation reactions are often energy liberating. I will say exergonic reactions. Whereas photosynthesis in the chloroplast is more in general a reduction, although it is mentioned as oxidation reduction both. But I am saying in more general it is reduction where energy is spent, not liberated. Clear? Energy is spent. So here I will say endergonic, just opposite. Okay. Nextly, the ATP synthesis occurs in mitochondria and in the chloroplast. That ATP synthesis is somehow related to the electron transport chain. Now that electron is actually provided by organic compounds in mitochondria and is pulled by oxygen. So oxygen dependent electron transport chain in mitochondria, which later resulted in ATP synthesis. So that's why formation of ATPs on the basis of oxygen dependency is called as oxidative phosphorylation. And when we are talking about ATP synthesis in the chloroplast, I repeat, ATP synthesis in the chloroplast, it will be photophosphorylation, means the light induced the oxidation of water molecule which liberated the electron, and the electron transport chain is actually driven by or induced by sunlight. So that's why light dependent ATP synthesis, here oxygen dependent ATP synthesis. So that's why the two are becoming different. In the inner membrane of mitochondria, we have studied certain stalked particle, means having head, neck and base. The particles are called as oxysomes in mitochondria. Where they are present? They are present on inner membrane, the foldings of inner membrane, cristae. This part is called as F1, this part is called as F0. Remember, the stalk is there. A similar type of structure is present on thylakoid membrane. I repeat, on thylakoid membrane. But that structure lacks the neck, means it is having the head, it is having the base. The head is mentioned as similar to F1 in function, but more correctly chloroplast F1, and this base chloroplast F0. F0, like in the two places, are going to function in the terms of proton channel, and F1 is going to be the enzymatic part. Both places same, but this is stalkless. This one is this is having the stalk. This is stalkless, right? Next, as the bacterial cells, let's compare about the bacterial cell here. So as bacterial cell, hetero and autotrophic. Why? Because here, as I told you, that they are liberating CO2 from the organic compounds. The one which breaks organic compounds to derive the energy are heterotrophic cells or the heterotrophic organisms. So if mitochondria is treated as a bacterial cell, then what kind of bacteria it will be? Heterotrophic or autotrophic? Answer is heterotrophic. It will be heterotrophic. Number 2, on the side of chloroplast, because it is deriving electron from inorganic compound, oxidation of inorganic compound, it must be autotrophic, right? So if mitochondria and chloroplast are considered as a bacterial cell, in that cases mitochondria must be some heterotrophic bacteria and chloroplast must be autotrophic bacteria. Altman proposed different names for the two structures. For mitochondria, he proposed the term bioplast, whereas for chloroplast, he called the word autoplast. Okay. So both of them are self-duplicating cell organelles. They have DNA in them. They are going to form their copies whenever required in the cell. So we have compared a good number of similarities and obviously dissimilarities, that how these semi-autonomous cell organelles are doing their work. They are different in the terms of work they perform, okay, but they both are considered equivalent to a bacterial cell, right? However, one is heterotrophic, other one is autotrophic. So this is the conclusion. So try to compare the things whenever they are going to end, so that you can retain most of the things about them. It is possible to retain everything. See, if the question is going to be asked as a direct line of the NCERT book, then there is no problem. Every student in the exam is going to attempt such questions. But when It is about deviated from the NCERT, a line which is not directly given in the NCERT. Such type of fundamentals are going to help you a lot. You can answer such difficult questions in a better way, or you can say in the correct way, because you have studied the fundamentals, you have studied up till the roots. Okay, if anyone is saying in-depth study, it is actually the study of fundamentals. This is the fundamental, this is logic based, everything is given in NCERT. We have just made those things, those lines into a table. We have collected everything from the NCERT itself. Okay, no matters that whether it is given in the cell as a basic unit of life or in the later chapter like respiration and photosynthesis. You need to have such kind of comparison wherever you find certain similarities and dissimilarities both in different entities or different features. Okay, coming to the next, today's cell organelle is ribosome. Now remember about ribosome, although we are studying the eukaryotic cell, but still I can mention the ribosome is the only cell organelle which is present in both prokaryotic and eukaryotic cells. Okay, these are present in both prokaryotic cells and eukaryotic cells. So this is how we can explain the things. Now moving to the next thing or the next features. What are the next feature about ribosomes? So ribosome, if they are present in the prokaryotes, and we know this thing that prokaryotes don't have membrane bound cell organelles, so it is very clear to us that ribosomes are also not having limiting membrane, lack limiting membrane. Okay, so this is how we need to mention about ribosomes. Moving next, about the occurrence of ribosomes. So where ribosomes are present, just now I told you the cell organelle ribosome is present in both pro and eukaryotic cells, but in eukaryotic cells the ribosomes are present in mitochondria and plastids as well, means in the cell organelles. Okay, so there are several types of occurrence of ribosomes. Secondly, we say if they are present in the eukaryotic cell, if we are going to consider a eukaryotic cell, then eukaryotic cell is having both type of ribosomes in them. One is called as 80S and the other is called as 70S. But before that, how the ribosomes are present or placed in eukaryotic cell? So they are placed either in the cytoplasm, clear, in the cytoplasm, or they are present in the cell organelles. About the cytoplasm, if I say, such type of ribosomes which are present in the cytoplasm, we call them as cytosolic ribosomes. Whereas those which are present in organelles are named as organellar ribosomes. So in this way I can mention the things very clearly. What? That the cytosolic ribosomes actually are of 80S type, and those which are present in the organelles are of 70S type. So here are the things: the cytosolic ribosomes 80S and organelle ribosomes, means which are present in where? Which cell organelles? Mitochondria and plastids in the organelles, in mitochondria. In plastids. So those who are in the mitochondria are called as mitosomes, and those which are in plastids are called as plastidosomes. So mitosomes and plastidosomes. Okay, ribosome is having so many names. When we were studying about rough endoplasmic reticulum, there I mentioned the name ergosomes. Now just I told you ribosomes. Now in prokaryotic cell we have studied them, either present freely, monosomes, or maybe several units are joined to single mRNA, polysomes, okay, plus who discovered ribosome. On that basis, they are called as pellet particles as well. George Pellet discovered ribosomes. So there are so many names of the same cell organelle. These many names. The cell organelle is same, right? So what about these 80S ribosomes? See, the 80S ribosomes are either present free in the cytoplasm, right? Free in the cytoplasm as monosome or polysome, although polysome are more common in prokaryotic cells. And the other one is on the cell organelle, on. Now this is in and this is on. Obviously the two things are different, right? The two things are not exactly same. On the table, the book is on the table, right? Means present on the surface. Which surface? Where these are present, where these 80S ribosomes are present. If they are on surface, we will say on surface of outer nuclear membrane and the surface of rough endoplasmic reticulum. Okay. So the cell organelle or the ribosome which is present on the surface of organelles are 80S, or 80S ribosomes may be present freely in the cytoplasm. The second thing, if they are present in the organelles, they must be 70S types. Okay. This is how we can mention about their occurrence. So many questions can be generated. Name the cell organelle which is present in both prokaryotic, eukaryotic. Answer: ribosomes. Name the type of ribosome which is present in both prokaryotic and eukaryotic. Answer: 70S ribosomes. Name the type of ribosome present in eukaryotic cell, both 80S and 70S. Name the type of ribosome which is present in organelle of eukaryotic cell and cytoplasm of prokaryotic cell, 70S. Which type of ribosome is exclusive to eukaryotic cell? 80S. Such questions can be created from the same fundamental thing, right? Next, for this type of questions, you need to do the practice. The more the practice you will do, more such questions come across you, and definitely you are going to be more confident in getting the correct answer. So practice is needed. For that practice purpose, we are putting up the daily practice paper along with the class. So it is recommended, highly recommended, that you must go through the DPPs and solve them, and thereafter appear for the weekend exam on the Yup Master. By this way, you are going to get highest score in your dream exam. Whether it's the exam in your schools or the exam of NEET 2021 or 2022. Okay, students, so we are going to proceed further. Let's see what next about the ribosome. Next is obviously about the structure. Now the ribosome is comprising of the two subunits. Okay, ribosome, as we say, they are discovered by Pellet, but their structure was not easy to study until the electron microscope was invented. After the invention of the electron microscope, we came to know that each unit of ribosome is consisting of two subunits. I said two subunits. And the two subunits are different because one is large and the other one is considered as small. So the large and small, these are the two different subunits in the ribosome. Although the two subunits will join at the time of function, means during protein synthesis, not earlier to that. So the two subunits differ in the S. S means S is related to sedimentation. During centrifugation. Now what does it mean? If we are churning, means we are moving the cream of milk, then that cream is going to separate into two parts. The heavier one, the butter, is going to settle towards the center, and the buttermilk, the liquid portion, is going outside. So heavier the material, earlier it will get sedimented, and towards center it is going to sediment. So the centrifugation is a method which is separating particles on the basis of molecular density. Centrifugation is separation of particles on the basis of molecular density. As I said, the one who is having higher molecular density, it will get settled down first. The lowermost layer in the centrifuge is obviously of the material which is more heavier. Then comes the light, then comes the lighter. Okay, the one which is going to settle first and the bottom is the heaviest one, or the molecular density value is highest. So molecular density is nothing, it is actually related to the size, or you can say size and the weight, or simply density in the size. This S value indirectly tell us about molecular density and size. The information that we are getting from S, Severberg constant. Okay, so here we are saying the two values are, or the two subunits are differing in the values of S. One is larger and the other one is smaller unit. So the two subunits, when they are going to join and how they are going to join, this we need to understand, right? This we need to understand. Here is the description, see. So the two subunits here. So here I am making the first subunit. This is the first subunit. Now this first subunit, I am saying this one is larger subunit. Whereas this is the smaller one. You can define their parts as well. Here this is called as base and this is the head. Here it is having a clefted part. Let's see. So the larger subunit is not this much straight. This is actually chair-like conformation, or you can say the chair-like structure. This is it. Okay, a three-dimensional type of structure. The small subunit is this. This is going to set on this one, and this is how it looks. But the two will join only at the time of protein synthesis under magnesium ion critical concentration. Means this value is specific. So the union of two subunits, or simply the stability of the ribosome structure, requires magnesium ion, and later it is going to look like this. Although this time my diagrams are little bit different from the NCERT, because we need to understand that how they appear, actually how they appear. So this is the thing that we can mention about the ribosome. The small subunit and the larger subunit. Okay. Now if I am going to consider them as ATS type, this base is going to attach on the surface of RER. Attaches to the surface of RER, or outer nuclear membrane. Okay, and why? It is due to a glycoprotein named as riboforin. Remember, this is about ATS types. Okay. Now the larger and small subunits are not same for the 70S and 80S ribosomes. They are obviously different. This is the functional form, and I told you that this will appear, this structure will appear when? During translation or protein synthesis. Okay, this is during translation or protein synthesis, right? So such type of thing we can mention about the structure of ribosome. Stability of structure of ribosome depends on magnesium ion concentration. Okay, the most important thing. The two subunits of ribosomes join under the influence of magnesium ion concentration. The two subunits of ribosome join at the time of protein synthesis, or at the time of function of the ribosome. Else they may be separated from one another, right? This is what you need to know, right? Coming to this. So this is what we studied about the structure. Now the next thing is about their chemical composition. Now as I told you, this ribosome means two different parts. Chemically, ribosomes are ribonucleoproteins, and if they are ribonucleoproteins, I am saying that this is having two things in it. The ribo means rRNA, ribosomal RNA, okay, or in short you used to write it as rRNA. And when it is considered about the rest of the part means the proteins, okay, this protein part, these are proteins. In terms of the function, what this rRNA is performing and what these proteins are performing. The function of rRNA can be one, structure of the ribosome, and number two, enzymatic during, or you can say catalytic during protein synthesis. And here we say about the proteins. What about proteins then? Here I am saying proteins are only of structural types. They are performing structural roles. Okay, got it. If this is the thing, then I need to know about that what type of rRNAs are there and how the large and small subunits are differing in the type of rRNA they possess. Definitely they are different. I am going to compare the two, means the 70S and 80S with each other. So how the two are same and different? Let's see. (70S) and (80S). It is not about their occurrence. Right now we are talking about their large and small subunit and their components, right? So the 70S type is having large subunit as 50S, small subunit or smaller subunit as 30S. Whereas on the other side the 80S is different. How it is different? Because the large subunit is 60S and the smaller subunit is 40S. One thing I learned from here, and that is what? The value of this total ribosome, the S, the sedimentation value, is not as per the addition of 50 and 30, is not as per the addition of 60 and 40. Why? Because we are saying the value of S are non-additive. I repeat, the value of S, the value of S are non-additive, or you can say non-cumulative. They are not supposed to be added up. Don't say 50 plus 30 is 80S. No. 50 plus 30, the two subunits are going to join to form 70S ribosomes. Their values are not additive. Now the next feature is about, or the next comparison is about the type of rRNAs they possess. This is more important rather than the type of proteins they have, because rRNAs are performing one structural role, second enzymatic role. That's why it's important. The rRNAs that are present in 70S, are 23S, then comes 16S, then comes 5S. Okay, and on the other side in the 80S, the rRNAs are 28S, 18S. In addition, they have one more thing, 5.8S and 5S. Okay, and obviously they have proteins, no doubt about it. So the composition is not same. That's why their molecular densities are also different. Okay, the number of proteins will also differ, that how many proteins do they possess. So this is the thing that we need to know about their chemical structure and the composition, that how the two subunits are going to be formed. Okay, the 70S is present in both pro and eukaryotic cell. The 80S is present only in the eukaryotic cell. The eukaryotic cell is having both 70S and 80S. Okay, the place may differ, right? Coming to the next thing, the origin. So, as I told you that the ribosome is going to be formed by protein and rRNA. As I said, ribosomes are made up of two things. What are those two things? One is rRNA and the other one is protein. So it's a very common thing that we know from beginning, and that is what? That proteins are synthesized in the cytoplasm on the ribosomes. So it means these proteins, we are talking about eukaryotic cell right now, okay? So proteins are synthesized in cytoplasm, obviously on the ribosomes, okay? And then the rRNA, obviously synthesized in the term, we haven't studied this term right now. This is nucleolus. In nucleus is fine. Okay. So it is like the one of the material is present in the cytoplasm already and the other is coming from the other side of the border. Border means I am saying from the nuclear membrane, crossing the nuclear membrane. The rRNA is crossing the nuclear membrane, coming to the cytoplasm where it get associated with the proteins to form the ribosome. Okay, so cross border, right? So this is how we are saying the ribosomes are formed from rRNA, which are made in the nucleus, particularly nucleolus in the nucleus, and the proteins are made by ribosomes themselves. So it's a good enough question to raise again, just like what you used to study in the lower classes or just in the beginning, that what comes first, the egg or the hen? The same is the question, what comes first, the proteins or the ribosomes? Because if we are saying proteins are synthesized in cytoplasm, they are synthesized on ribosome. The proteins that are making ribosomes are synthesized by ribosomes. Interesting. Okay, obviously it is ribosomes that came first. So this is how we are saying about the origin. So ribosomes are not self-duplicating, remember. Okay, although I can say one thing, if cell is actively participating in protein synthesis or cell is more interested in doing protein synthesis, then they will have more number of ribosomes in them. Okay, so their occurrence is going to be affected if the cell is doing protein synthesis. If the cell is not related to protein synthesis, they will have fewer number of ribosomes in them. Number of ribosomes more in the cells, which are participating actively in protein synthesis. Obviously ribosome is the protein factory of the cell. This is it, right? So here we concluded about the origin or their numbers. Next, about the function of ribosome. One enough function is there, and that is what? The ribosomes are protein factories of the cell. They may be observed as some modified structures, particularly in the neurons. We call them as Nissl granules. They are nothing, they are actually what? These are RER and ribosomes. In the cytoplasm of neuron. Or nerve cell. A modified structure. So this is how we have compared the two things, right? So whenever it is asked that what function they are performing, means ribosomes are performing what? Protein synthesis. One enough good number of function, one only, that's it. So next is about the other structure which don't have membrane, but they are going to support the membrane. Okay, let's talk about that function, that structure actually. So the eukaryotic cell is having extensive membrane. See this, this is the endoplasmic reticulum. Okay, and this is what we are saying Golgi. Now one more thing is there in this eukaryotic cell, and that is about movement of cytoplasm around the cell organelle first, or maybe in the whole of the cell. Now that function is called as cyclosis or cytoplasmic streaming. So when the cytoplasm is moving, although slowly, or you are saying intracellular circulation is taking place within the cell, there is a chance that these cell organelles may get collided to each other. This collision is to be avoided. Okay, so how the collision is avoided? There must be something or some frame like structure which is avoiding the collision of the cell organelles. Now that framework is given by cytoskeleton. It is just like, say for example, if the room is having walls, the structure will become more and more sustainable or more stable if the walls are getting separated, in particular by the pillars and the beams. The pillars and the beams are maintaining the structure, keeping the walls in the place. Just like when we are going to rise a tent, the tent is getting support by bamboos which are acting as pillars, the ropes which are going to give tension to the structure, and this is how the structure is becoming more sustainable or more stable. So the same thing in the eukaryotic cell is about the structure or the membrane, extensive membrane in the eukaryotic cell. And two, the second type is keeping the cell organelle in the place. Now it is all possible because of the cytoskeleton, right? Means we are saying that internally the eukaryotic cell is having a framework, protein framework. The protein framework in eukaryotic cells. And therefore you can say what is the function of this structure. So I can say simply that this framework is going to keep cell organelles in place, okay, and second, keep the cell organelles in place, and the second function what it is going to perform is support to the membranes, and so many functions are there. We are going to learn about the functions, we are going to know about the role of cytoskeleton. Even this movement of cytoplasm in the eukaryotic cell, the cytoplasmic streaming or the cyclosis, also because of cytoskeleton here. So this is how we need to know about, right? So whenever it is asked that where cytoskeleton is present, answer is eukaryotic cell. Okay, they are not present or not seen in prokaryotic cell. The prokaryotic cell is having a simple structure. They need not to have that kind of framework because they don't have cell organelles in them. They don't have extensive membranes inside. So keeping the cell organelles in position and maintaining the membrane structure, this cytoskeleton is going to be important here. Now that cytoskeleton, which is actually the protein framework, is consisting of different type of component. But before that you need to know about certain fundamental things. Now compare the things which I am saying about that. If I have a car, just like a simple car, I am saying this car so the tires are not much important here, the other things are more important. If I am going to look for the different components in the car, I will say different components in the car, I will say this is the bumper portion and is going to be solid. Thereafter comes the engine. This is again solid. In between is a hollow space. Okay. then again the hollow space, and then again the solid space. Why so? So I have two type of component. I am saying hollow and I am saying the solid parts. Now this is going to give the car a structure which is going to sustain any kind of damage or the collision. See how? See, for example, if the structure is solid, it is helpful in damaging the other structure. And if the structure is hollow, it is going to absorb the force. So this driver is going to have lesser impact. Say for example, if any accident took place, bang, then the impact is lessened by these hollow parts. So hollow parts are something which are called as compression bearing. I repeat, these hollow things are what? These are compression bearing. It is just like that a person jumped from the top of the building, 8th floor, fall on the ground. What will happen to that person? Obviously, the body is going to rupture, the bones are going to be broken down, the person may die. But person jumped, stuntman in a film, jumped from the 8th floor. Below on the ground they had kept a number of cartons and some mattresses, other things. Now the person when jumped, he is having more surety to survive. The reason is, whenever the force is considered, that force is going to disperse because of the hollow boxes or the hollow cartons. Okay, so we are saying that compression is what? It is actually distributing the damaging force. And the solid is going to give tension, means it is creating the impact. It is going to help in putting the force on the other objects. So this is why we say the car structure is having both the type of thing, compression and tension. So the framework consists of both things, remember. Any framework, if it is required to be more safe, it must have compression and tension bearing components. The same is about cytoskeleton. In the eukaryotic cell, these proteinaceous filaments are going to be of three kinds. The one is microtubule, which is obviously the longest or the largest of all. It is made up of protein which is called as tubulin, but is hollow cylinder. So what I can tell about, or what I can say about microtubules, they are compression bearing. Okay, the microfilaments, as the name says, they are small. They are smallest actually. Intermediate filament are second to the number. Now microfilaments are made up of what type of thing? They have something contractile proteins like actin, myosin type of components. But these are solid. So you can understand how they are going to work. The intermediate filaments are something what? They are intermediate, means in between the sizes of microtubule and microfilaments, they are made up of structural proteins. The structural proteins which are like desmin, vimentin, etc. And they are also solid. Tension bearing, compression bearing. The microtubules are important. These microtubules are going to form different structures. What structures? We are going to study about the microtubular organization only, not about the microfilament and intermediate filament. We will study only about their functions or where they are present, what they are doing. But microtubules we need to study a lot. We need to study that what microtubules forms, when these structures are formed and what they are doing. The microtubules are going to give rise to or organize into a type of cell organelle. What? Those cell organelles are locomotory structure, cilia, flagella, or maybe non-locomotory structure like centrosome, basal body. Now the same microtubule is making what? Spindle fibers or spindle apparatus. Spindle apparatus means the structure which helps in movement of chromosome during cell division. Okay, so these all are the functions of microtubules. This microtubule only determines that where the cell plate is going to form in the plant cell, or the cell furrow is going to be formed because of microfilament. Now different means the functions related to plasma membrane are because of microfilament. Supports plasma membrane. The pseudopodia or the false feet in amoeba is because of microfilaments, false feet or pseudopodia in amoeba. Okay, these all are the things which are performed by microfilament. The intermediate filament is going to form the nuclear lamina, means a structure which is inside the nuclear membrane. We will study about it in the nucleus, in the diagram of the nucleus we will label it. This is going to form nuclear lamina in certain animal cells, not in the plant cells. So obviously we are going to do the details of microtubular organization into cilia, flagella and centrosome basal bodies. The function I told you, the microtubule are going to form these many structures. The microfilament is going to support the plasma membrane, forming the false feet, doing the cytoplasmic streaming, etc. The intermediate filaments is the structural one, forming the nuclear lamina in the nucleus of the certain type of animal cells. So these are the functions. So the next which we are going to learn about is microtubular organization, the cilia, flagella, centrosome, basal body. That will be the separate part. What we learned about the function of cytoskeleton, just I said in common, I can say the cytoskeleton is providing the cell organelles their actual place and avoiding their collision during cytoplasmic streaming. The cytoskeleton is providing the strength to the membranes and keeping them in the proper position. The cytoskeleton is related to cytoplasmic streaming. The components of cytoskeleton are going to provide the cell organelle related to locomotion, cilia, flagella. Okay, give rise to the cilia, flagella, basal body. Give rise to the ester in animal cells, centrosome. Okay, and the microfilament, other functions, intermediate filaments, other functions. This is all for today's class. We are going to start with the next microtubular organization type of things in the next class. Means we are going to study the locomotion due to cilia, locomotion due to flagella. And we are going to compare cilia, flagella with basal body and centrosome. Then how they are same, how they are different. We will study about the axoneme and the cartwheel structure in the very next class. Before that, you are advised to attempt the daily practice paper so that you can improve your grasping, you can improve the efficiency and the accuracy of the topic that has been told in today's class. Be regular in your studies and enjoy the learning only on Yup Master to achieve the best rank in NEET 2021, NEET 2022. Thank you for watching the classes. All the best.